Thank you very much. And I think now we should move to the last lecture in the series. That is by Professor T.V. Hande, who is from Shri Shivaji Science College, Amravati. He is mainly working on the endophytic fungi, and he did his PhD from Amravati University only. He is presently professor in the Department of Botany, and he has produced two PhD students, two MPhil students, and 5 are still working under his supervision. He has attended many national and international conferences and has been awarded with the Dr. B.R. Shobe Memorial Lecture in 2017. With this brief introduction, I would like to invite Professor T.V. Hande to deliver his talk.
Professor Hande, thank you, sir. 20 minutes, please. Yes, sir. Yeah. Sir, may I audible?
Yes, you are audible.
Thank you, sir.
Slide is visible.
Honorable Chairman, Dr. Gurupal Singh, Honorable Co-Chairman, Senior Mycologist of MSI, and all MSI members. I am presenting my topic, Endophytic fungi from medicinal plants, the source of bioactive secondary metabolite. So everybody knows the endophytes. These endophytes are growing inside the host without any symptoms. They are asymptomatically. Most of the scientists who are coining the term endophytes are any organism occurring within the plant tissue and other things. So these are the different mycologists and scientists who coin the term. These endophytes are very important, which is having the multifunctional role of the fungal endophytes in plant tissue. These fungal endophytes, which are having the specific type of the metabolites, these endophytes interact with the plant metabolism. They are giving interaction with the plant pathogens and interaction with the herbivores. These endophytes represent a complex community of microorganisms colonizing asymptomatically internal tissue of the plants. These endophytes, which are very important because they are producing the anti-cancer agents, antimicrobial agents, and so many industrial products. These endophytes enhance the fitness of the host plants by direct production of bioactive secondary metabolites, which are involved in protecting the host against herbivores and pathogenic microbes. These are approximately, it has been estimated that more than 1 million different endophytic fungal strains inhabit about 3 lakh variant plant species. In addition, the fungal endophytes are useful resource for natural products which effectively have advantage of in agriculture and the industry. The uniqueness of the endophytic fungi as alternative source of pharmaceutically valuable compounds originally isolated from the higher plants, fungal biosynthesis of the plant associated with fungal endophyte metabolites, as well as new approaches developed to improve the production of commercially important plant-derived compounds with the involvement of endophytic fungi. For example, the muscador is a novel endophytic fungal genus that produces bioactive volatile organic compound. Moreover, the hydrocarbon profile of endophytic fungus Biocladium luteum was discovered with the various compounds associated with diesel fuel, that is the mycodiesel. All these fungal metabolites are classified together as a secondary metabolite. The term secondary metabolite denotes the biochemical process underlying the formation of fungal metabolites. Most secondary metabolite synthesis by endophytic fungi are not chemically characterized, but they have remarkable importance with reference to mankind. Gas chromatography can separate the compound of the volatile and semi-volatile nature of reticulocytes. On the other hand, mass spectroscopy can identify the compound with great efficiency. Characterization of structure and conformation of the identity are usually achieved by comparison of the mass spectra, molecular spectra, and determination of chromatographic detection index. So the इसको methodology I have selected these medicinal plants because they are very important medicinally, which were collected from our forest of the Amravati district, the map of the Amravati district. The collection of the plant material which are being carried out by the standard methods, then the isolation of these endophytic fungi which are also carried out by the standard methods, and the surface sterilization and other things all are carried out. The fungi obtained on this media which was identified on the basis of morphological identification with the help of standard taxonomic keys and monograph, and the colonization frequency of fungal endophytes were calculated by using the following formulas. This is the general type of the extraction of the method of the fungal extracts which we have carried out. That is the culture and filtration, culture broth, and the extract which was used for the further analysis. Then the antimicrobial activity have been carried out by the standard method, which is the TPD assay. It is were prepared and which was forwarded for the antimicrobial activity. Software or text or the agar plates which are used and were placed for the incubation and the inhibition zones, which was recorded. The antifungal activity was determined by measuring the diameter of inhibition zone in millimeter. Then the gas chromatography mass spectroscopy have been carried out for the analysis of the sample and sent to the IIT. Then identification of compounds have been carried out at the
slides are not
database.
visible. Your slides are not visible.
Slides are not moving.
Your slides are not visible, please.
Just a minute. Now visible?
No, nothing.
Okay. Now it is visible?
No.
You have to, I think, rejoin again, and that is why it is struck
I
up.
will share and again join the share. Again share.
Dr. Hande, the slides were visible. Project it again, yes.
Yeah.
Project again. Go to slideshow.
Go to the slides. Move ahead.
Yeah.
It is visible, sir.
Yes, yes.
Now it.
Okay. Then in case of observation and results, the Arthenium duoigespermum is the endophytes which are isolated, and on the basis of their morphological studies, we have identified this fungus, that is the Conidia sessile, or sometimes short highlighted peg-like slide sides conical pores which are having the spores. This is the Arthenium Hande, in which the characteristic feature of this fungus, that is the Conidia brown unisect globose lenticular with equatorial slit. This is the next endophytic fungus, Epicopum nigrum, which are isolated from these medicinal plants, and the Conidia are non-septate, rounded, daily color, and the gangliospores are present. Fusarium igesforum, which is a very common fungus, which are isolated from the endophytes, the microconidia and macroconidia are present. This is the Nigrospora moraiji, which is having the conidia borne singly at the tip of the conidia pores, globose or somewhat flattened. This is a Blastopsis frintaria, which is having the conidia which are broad, tapering toward the base and clavate reside straight with 5 set, which is a very important endophyte isolated. This is one is the Pithomyces chartarum is a endophyte which is isolated from the plants, that is which is having the characteristic feature, that is the conidia produced singly and the blood out involves ovoid, elliptical, pale brown and end stage. This is the next endophyte, that is the Stachypodric chartarum isolated. The Stachypodric chartarum, which is having the conidia smooth single oval at young stage and elliptical when mature, dark green, black. The next endophytic fungus, that is a Stachypodric nilgrica, which was identified on the basis of morphological characters, that is the conidia borne singly are acrogenously at the tip of the phthalites, pulsate and globose. This is a Trematospora uzi is the endophyte which is identified on the basis of morphological characters. Conidia have only one longitudinal septa. These are the cultural characteristic feature of these endophytes we have identified, isolated from the medicinal plants. In case of diversity of endophytic fungi, it is observed that the diversity of endophytic fungi were isolated from medicinal plants collected from the forest and samples which were collected in the three different seasons, and these are the diversity of this fungi which is studied on three different seasons, that is the monsoon, winter and summer. And of the three season, it is observed that the percentage occurrence of this fungi, colonization frequency of this endophytic fungi, which is more in the monsoon and the winter. In case of the different medicinal plants like Calotropis procera, these are the diversity of the endophytic fungi which are isolated, and again the diversity and the percentage occurrence of these endophytes which are more in winter and the monsoon. In case of the plant, medicinal plant which is very important, that is the Cissus quadrangularis, Cissus quadrangularis, which are showing in the endophyte diversity in the monsoon and the winter season in comparison to summer season. So in case of the next the plant, that is the coffee arabica, which is showing the endophytes diversity in monsoon and the winter season, and this diversity again is studied in the different part of the plants, that is the stem, leaf and the petiole, which shows the variation of this fungal diversity. This is the next plant, that is the datura tomato, showing the fungal diversity and their percentage frequency in the different season, and from this table it is noted that this summer season which is lacking the diversity of fungal endophyte, then the fungal endophytes isolated from this Diasporia bulbifera, which is having the fungal diversity in the winter season. This winter season is favorable for the fungal endophytic development. And in case of DILIP V, Isola, the fungal diversity found in the winter season. And in case of heavy decimus, the fungal diversity is more in the monsoon season. In case of Uraya, the fungal diversity which is found in the monsoon and the winter season. And in case of Osimum, this diversity is all around the year. So these are the fungal diversity of the fungal endophytes and percentage frequency of the fungal endophytes in the different medicinal plants. Then in case of antimicrobial properties, antibacterial potential of isolated endophytic fungi were tested against selected pathogenic bacteria. These three bacteria which were used, that is the Escherichia coli, Staphylococcus aureus and the Pseudomonas aeruginosa, which were learned from the MPCC. So these are the activity of this fungus aspergillus tinctorius against Escherichia coli, which are showing the inhibition zone. Nigrospora oryzae against the Pseudomonas aeruginosa. This is the antimicrobial activity of the Arcenium phoeoideosporum, which are showing the zone of inhibition. This Alternaria alternata showing the zone of inhibition against the Pseudomonas aeruginosa. Antibiotic charterus, which clearly zone against the Pseudomonas aeruginosa. In Nigrospora oryzae and Nigrospora oryzae showing the inhibition zone on both the bacteria, against both the bacteria, that is the Pseudomonas aeruginosa and Escherichia coli. So these are the antimicrobial activity and inhibition zone against the different type of the bacteria and from the fungal endophyte. This is the Nigrospora oryzae and shows the antimicrobial activity against the Escherichia coli. Arcenium Hande is the endophyte. This is Stachybotrys chartaris, which shows the antimicrobial activity. Trematostoma carularia, this endophyte which shows the antimicrobial zone against the. Then after antimicrobial activity, the DCMS have been carried out. This is the chromatogram of the DCMS, and this chromatogram which shows of the Arcenium fungus showing the peak and retention time. These are the identified compounds present in the Arcenium phoeoideosporum extract. These are the structure of compounds. This is the chromatogram Nigrospora oryzae showing the peak with the retention time. These are the compounds identified from the Nigrospora oryzae. These are the structures of these compounds. The chromatogram, this is the Trematostoma hughes showing the peak with the retention time. These are the compounds which are identified from the dermatostoma using. These are the structure of chromatophore. Chromatogram of the epithelium nigrum showing the peak with retention time. These are the compounds identified from the extract of epithelium nigrum. These are the structure of compounds identified in the sample. This is a chromatogram of the static body DILIP showing the peak with retention time, and these are the compounds, these are the structure of the compound. Then after this the FTRI analysis have been carried out, and during the FTRI analysis, the FTRI analysis of arsenium fluoigespermum extract, this arsenium show the presence of sulfonyl alkyl amine group on the basis of this peak and the wave number which was recorded. In case of the Stachybotrys nigricans fungus endophyte, they on the basis of this FTRI analysis, the sulfonyl and amine functional groups are detected. Then in case of Stachybotrys chartarum, on the basis of this peak and the wavelength, the sulfonyl alkane and amine group is detected. On the basis, in case of the Nigrospora oryzae endophytes, the effect of Nigrospora oryzae endophyte, which shows the on the basis of the absorption tetra of the crude extract of this Nigrospora oryzae, which is having the sulfonyl and amine functional group. So this is the FTRI analysis of the Epicocum nigrum, which is having the alkene and amine functional group, and this is the analysis of the Pestaloeopsis funereum, is the species endophyte which is having the carbohydrate and amine group. So in the last part, that is the conclusion. In the conclusion, morphotaxonomical study of endophytic fungi is very essential to establish the diversity. During investigation, anamorphic fungi were predominantly occurred. Most abundant species are the Alternaria, Aspergillus, Cladosporium, and the Pestaloeopsis. The endophytes show higher colonization frequency in monsoon as well as in winter as compared to summer. Conclusively, it suggests that environmental factors play an important role in the establishment of endophytic fungal diversity. Seasonal variation may cause the tissue specificity in colonization of endophytic fungi. Some species like Dipomycin chartarum, Stachybotrys nigricans, Trematostoma hugi isolated from the Calotropis, Procera revealed the tissue specificity in their colonization. These isolate colonize only leaf and the petiole tissue of this host. There is increasing the demand of bioactive metabolites that can be used in the industries, medicine, and agriculture. Endophytic fungi, often in the present study, exhibited great antimicrobial activity against the pathogenic microbes like Pseudomonas aeruginosa, Escherichia coli, and Staphylococcus aureus, which can be potential source for synthetic drug and other useful bioactive products. Endophytic fungi are a good source of bioactive secondary metabolites. The fungus, namely Arsenium Fusarium, isolated from Whiting's negundo, produce the antimicrobial compounds like the hexadecoxyl carbonyl, and these are the compounds which are very important for the industries which are isolated from this fungus, that is Arsenium Fusarium. It is noted that some compound has used in the industrial products. Ecosine and hexadecine is used as a lubricant and lubricant additives. Hexaponic acid used to produce the soap and cosmetics. Naphthalene mainly used as a precursor to other chemicals. Naphthalene, sulfonic acids are used in the synthesis of 1-naphthol and 2-naphthol, which are precursor for various dyestuff pigments, rubber processing chemical, and other pharmaceutical chemicals. Naphthalene also used as a household fumigant. Endophytic fungi are a novel and important microbial resource for producing bioactive compounds that have attracted the attention of many researchers worldwide. The future of discovering industrial products from endophytic fungi in harsh environment is undoubtedly bright. So, thank you very much. I am highly grateful for this great honor to Mycological Society of India, Secretary and all office bearer, and Department of Botany, Punjabi University, Patiala, all senior mycologists and MSI life experts. Thank you very much.
Thank you very much, Dr. Hande, for your presentation. Any questions from the members who are listening to the talk?
We are running short of time, sir.
Okay, then we are running short of time. So we did not have any questions. So I would like to thank Mr. Hande for his.
You can pass it on to Dr. Hande and he can answer later on. There is some question.
Yeah, that will be excellent. Any
Interesting.
question?
Talking on endophytic fungi, so there are many questions.
Yeah.
So there should be time for answering these also.
We will answer on the mail.
Yes, Dr. Hande can reply them on the mail or can reply them. That is very
that will be excellent idea.
One minute, sir. I request all the fellowship awardees to send their full-length papers to the editor-in-chief, Professor Yaspal Sharma, so that it will be published in the coming June issue. Thank you.
Yes, sir.
So I would.
Thank you. And congratulations, Dr. Hande, and namaste, Dinga sir. Good to see you.
Yes.
Namaskar.
Thank you. So I'll take this opportunity to thank Dr. Hande for his presentation, and I will also request Dr. Geeta Somli to comment on the lecture of Dr. Hande, please.
Well, the lecture was well made on endophytic fungi, and he has isolated so many endophytic fungi from a very large number of medicinal plants. He has been able to characterize some of the secondary metabolites also that have been produced by these endophytic fungi. But there is just one thing I would like to interact for just a minute only, and that is that he has been able to find out many such fungi which are present in the nature as saprophytes also. So, has he been able to characterize such isolates and compare them with the isolates which he has found as endophytic fungi? Because otherwise, if those saprophytic isolates also produce the same secondary metabolites, then there is no need to isolate them from the plants itself. We can get them from the nature as saprophytes also. So this is something which I would request Professor DILIP Hande to look into this matter, because he has found such fungi like Picoccum, Nigrospora, Stachybotrys, and they are very commonly found as saprophytes also. Otherwise, it is a very good, well-made, this presentation, and it was given very nicely by Dr. DILIP Hande.